So in this subtopic, we are going to study the definition of recombinant DNA technology and also the 5 tools used in this technology. Okay, basically, you should know their function for target DNA, which contains the gene of interest, and the restriction enzyme, and also the vector used to transfer the DNA into the whole cell. And we have the whole cell, and also the modifying enzymes. Sebelum kita faham tentang recombinant DNA technology, kita faham dulu apa maksudnya recombinant DNA. Recombinant adalah maksudnya gabungan semula. Sebab sekarang DNA ni adalah hasil daripada gabungan DNA dengan gene daripada spesies yang berbeza. Dan DNA ni kita panggil dia foreign gene. Foreign, berbeza atau luar. Okay? So, biasanya dia adalah asal daripada spesies yang berbeza. Okay, dalam silibus kita, recombinant DNA yang paling biasa adalah plasmid. So, kalau refer balik chapter 2, plasmid adalah satu DNA yang dalam saiz kecil, bentuk gelang atau we call it circular and double-stranded in the cytoplasm of the bacterial cell. Okay, so if you refer back to your lecture note in chapter 2, you will notice that originally they are in circular shape. But now in this chapter, we notice this plasmid able to be modified and accept the foreign gene from other species, okay, which is represented by the green color here. And when they are ligated by the DNA ligase, they will become a recombinant DNA. Recombinant DNA technology is a method when the scientists combine the DNA from two different sources or even more than two. Yang penting usually organisme yang terlibat adalah berbeza spesies. Dan metod ini hanya boleh dibuat di luar badan hidupan. That's why we call it as in vitro. In vitro adalah istilah khas dalam bahasa Latin. Maksudnya, it's not done inside the body of living things. So, dengan senang kata, hanya boleh dijalankan di magma sahaja. Teknologi ini semakin penting sebab dia mempunyai beberapa tujuan yang boleh memajukan bidang sains. Yang paling pertama ialah saintis boleh menghasilkan bahagian DNA tertentu sahaja yang mengandungi gene yang mereka perlu dalam kuantiti yang banyak tetapi masa yang singkat. This is called amplification. Tujuan kedua, saintis dapat memperkenalkan DNA yang berbeza spesies ke dalam organisme tertentu atau sel tertentu. Dengan itu, jika organisme itu telah menerima DNA atau gene yang berbeza daripada asalnya, dia dapat menunjukkan phenotype yang baru. This is why we say novel functions. Novel maksudnya baru. For the second learning outcome in subtopic 7.1, students should be able to list 5 tools that use in recombinant DNA technologies. So basically, you should also understand the function of each of it. And we notice that there are 2 DNA involved here, which is the target DNA and the DNA cloning factor. And 1 cell involved, we call it as host cell. And there are 2 enzymes, or we say 2 types of enzymes involved, which are restriction enzyme and modifying enzyme. Target DNA is defined as a fragment of DNA containing gene of interest to be cloned. Mengapakah sesetengah DNA dilabel sebagai sasaran? Sebab dia mengandungi atau membawa gene yang diperlukan untuk tujuan tertentu. Gene itu sebenarnya adalah gene of interest. Dan kami akan pilih target DNA ini untuk tujuan cloning. Cloning maksudnya buat salinan. The next two is restriction enzymes or restriction endonuclease. Restriction ada maksudnya cutting atau memotong. This is why you look at the definition is an enzyme which cuts DNA molecules and produces short restriction fragments. This is why we call it endonuclease sebab dia boleh memotong nucleic acid. Okay. The main characteristic of restriction enzyme is they are specific. Because different restriction enzymes able to recognize different restriction site. So what is meant by restriction site is actually the specific and the short base pair sequences found in the DNA. So when a specific restriction enzyme able to recognize the restriction site, they will start to cut the phosphodiester bond to open the DNA strands. Restriction site yang dikenal pasti oleh restriction enzyme bukan sahaja they are short base pair sequences, but they are also palindrome, or we say they are palindromic sequence. So apa maksudnya palindrome? Means that if we read the basis sequences, right, they show same sequence from both opposite direction. Because the DNA molecules are double-stranded molecules, means they have the two strands which arrange in anti-parallel form from 5' to 3', and another complementary strand is 3' to 5'. But if we read the basis sequences for 5' to 3', the reading is GAATTC, and from the complementary strand, 5 to 3 also has the same reading. This is what is meant by palindrome. Always remember that restriction enzyme will cut DNA at the phosphodiester bond, but the way they cut will produce different types of cut ends. The first way is stacker way. When the restriction enzyme recognize their restriction site, they will cut in a zigzag way and produce a DNA with sticky ends. Why is sticky? Because now the cut open DNA, they have the unpaired nucleotide bases. They have the potential to complementarily base pair with another DNA fragments. Another way is straight way. When the restriction enzyme identified their restriction site, they will cut in a straight line and produce the DNA with blunt ends. Blunt ends means now no unpaired nucleotide base pair shown in the cut open part, or we say no overhangs found. So we are going to study two important examples of restriction enzymes that produce different cut ends. EcoR1 is the example of restriction enzyme produce sticky ends, and SMA1 is the example of restriction enzyme that produce blunt ends. Okay, jika anda perhatikan cara kita type nama EcoR1, Eco dalam keadaan italic because we wish to highlight the source of this enzyme. So scientists akan extract enzyme EcoR1 from the bacteria E. coli, Escherichia coli. And this RE or this enzyme only able to recognize from 5' to 3', which is GAATTC. And they will cut in a zigzag way between the guanine and adenine from 5 to 3. Complementary strand also, they will cut between the guanine and adenine from 5 to 3 because restriction site is palindrome. So now the cut open DNA will show us the sticky ends. However, SMA1 we notice that SMA is typed in italic form. Again, just to highlight the source of this enzyme is from bacteria Serratia marcescens. This restriction enzyme will recognize the base sequence from 5 to 3 in CCC, GGG, and they will cut a straight line in the middle of the restriction site, which is between the C and G from 5 to 3. One more extra example of restriction enzyme that can produce sticky ends is BAN-H1. So again, BAM was typed in italic form to highlight the source of this restriction enzyme, which is naturally produced by the bacteria Bacillus amyloquifacin, and they will identify the base sequences in 5' to 3' in terms of GGA TCC. So they cut in a zigzag way between the guanine from 5 to 3, same goes to the complementary strand, and produce also the sticky end, which always means the unpaired nucleotide bases. Cloning vectors is defined as a small DNA molecules that can carry foreign DNA fragment into a whole cell and replicate in that cell. So definition dia sebenarnya telah ditunjuk dalam nama dia tersebut. Vectors. Vectors ada maksudnya carrier, pembawa. So sekarang DNA molecules yang kecil ini boleh membawa foreign DNA daripada sumber berbeza ke dalam satu whole cell. So dalam whole cell ini, dia boleh menghasilkan banyak salinan foreign DNA tersebut. That's why nama dia dimulai dengan perkataan cloning. There are so many types of cloning vectors, which are plasmid, cosmid, and the YAC. These three are actually DNA molecules. But bacteriophage is actually a virus. So each of the cloning vectors here have their typical examples, or we say the popular examples. Okay, what makes them different from each other is actually related with their insert capacity. Insert capacity means that what is the size of the foreign DNA they can carry. So for example, plasmid is the cloning vectors that can carry 10 to 15 kilo base pairs of the foreign DNA. And compare with the YAC, which seems like the cloning vector can carry the largest foreign DNA, which is 100 to 1000 kilo base pair of foreign DNA. We have to depend on the scientists what type of the cloning vector they need to use by studying the size of the foreign DNA they wish to insert into the whole cell. In our syllabus, we are going to focus on plasmid only. Plasmid is defined as a small circular bacterial DNA carries the accessory genes separate from the bacterial chromosome. In chapter 2, we already studied in prokaryotic cells such as bacteria, there are two types of DNA found in the cytoplasm. One is the larger size, we call it as chromosome. Another one is the small in circular form, we call it as plasmid. So the popular example of plasmid used as a cloning vector is PUC18 that carry the accessory genes, means the genes of ampicillin resistance and also the LAT-Z gene here. Why a plasmid for particular species of bacteria can be used as a cloning vector is because they have these all four main characteristics. Firstly, cloning vector must be able to accept foreign DNA in multiple cloning sites. So, jika kita tengok contoh plasmid, multiple cloning site atau singkatan dia MCS sebenarnya merupakan sebahagian kecil dalam plasmid itu. So, we call it as short segment of the DNA. So, at the MCS, there is a list of the nucleotide base pair sequences. And at here, there are so many restriction sites can be identified by different restriction enzymes. So when the plasmid meets with the restriction enzyme, which able to cut the phosphodiester bond and open the plasmid and make it able to accept the foreign DNA. So jika kita tengok contoh gambar ini, maksudnya plasmid ini dia merupakan satu pilihan yang baik sebab dia mempunyai banyak pilihan untuk menguruskan penerimaan foreign DNA. Jika kita tengok restriction site dia, pilihan dia banyak. Sebab dia ada restriction site untuk band H1, dia ada restriction site untuk EcoR1 dan juga HIND3. The next characteristic of a cloning vector is they're able to replicate freely or independently in host cell. Plasmid, or the example PUC18, they have the origin of replication, which is written with the abbreviation ORI. ORI means a sequence of nucleotide base pair that initiate the DNA replication. Like what we have studied in chapter 5, for eukaryotic cell, the ORI is important to be recognized by helicase to start the unwinding for the DNA replication. So for bacterial cell, they have the plasmid, and the plasmid has the ORI, which is very important to give instruction for the plasmid to start the DNA replication independently in the host cell. Cloning vector such as plasmid, they has the accessory genes they always use as a genetic marker. So the example such as ampicillin resistance gene is used as the genetic marker during the screening method later. When a host cell is inserted with this plasmid that consists of the ampicillin resistant genes, the host cell is able to survive in the medium that contains antibiotic ampicillin. Or it means that the host cell cannot be killed by this antibiotic. The last characteristic of a cloning vector is the ability to express or amplify the clone gene under suitable conditions. Sebab cloning vector yang telah berjaya menerima foreign gene iaitu DNA yang isolated daripada organism which is different species, the cloning vector will be called as recombinant plasmid. Apabila recombinant plasmid ini dimasukkan dalam satu host cell atau sel perumah, dia mesti berjaya menunjukkan keupayaan menghasilkan produk mengikut gene yang dibawa itu. This is what we say express. The concept is similar with what we have studied in chapter 5. The genes can be expressed through transcription and translation process by producing the proteins that determine particular phenotype. Selain itu, cloning vector yang telah membawa foreign gene ini atau recombinant plasmid mesti ada keupayaan menghasilkan banyak foreign gene tersebut melalui DNA replication, atau kita panggil proses itu amplification. Host cell is defined as the cell that receives the DNA and able to replicate it. Maksudnya, sel perumah bukan sahaja perlu menerima recombinant DNA yang membawa foreign gene yang bukan sama spesies dengan dia, tapi dia perlu membekalkan satu keadaan yang optimum untuk DNA menjalankan DNA replication. Dan dalam bidang recombinant DNA technology, bakteria masih merupakan pilihan yang paling biasa sebab plasmid atau DNA bakteria ini boleh diasingkan dengan senang dan juga boleh dimasukkan semula. Dan apabila recombinant DNA ini reintroduce back into the bacteria, they're able to undergo binary fission, which is a type of cell division that the bacteria can produce many copies of the DNA. So examples of the bacteria that use as a host cell, which is the most common one, still E. coli. So the E. coli that carry the recombinant DNA, we call it as genetically engineered E. coli. While a bacterium can be chosen as a host cell in recombinant DNA technologies, actually depends on these three characteristics. First, they must be able to receive recombinant DNA through transformation process. After the resection of the recombinant DNA, they're able to maintain the structure of the recombinant DNA from one generation to another generation. This is because a host cells they must be able to amplify the gene product. This means they must be able to make many copies of the foreign genes through the binary fission process. So from one generation to another generation, the structure of the recombinant DNA should be maintained to enable the DNA replication, the transcription, and the translation process as well. DNA ligase is a type of modifying enzyme, which is a tool required in the DNA recombinant technology. The function is very similar with the DNA ligase involved in DNA replication because they are going to catalyze the formation of phosphodiester bond. When the plasmid and the foreign DNA are cut by the same restriction enzyme, they will be treated with DNA ligase to be joined as a recombinant plasmid or recombinant DNA. That polymerase is another type of modifying enzyme. That TAQ was typed in italic form to imply the origin of this enzyme from the bacteria Thermus aquaticus. Their function is very similar to the DNA polymerase class 3 we studied in DNA replication, which catalyzed the addition of DNA nucleotide to the 3' end of primer. The main difference between Taq polymerase with DNA polymerase class 3 is they are thermostable or heat-stable polymerase since that they are isolated from the hot spring bacteria. And this characteristic makes this enzyme a better choice by scientists to be used in PCR, polymerase chain reaction, because one of the steps will be done in high temperature.